Hello there to you in Amsterdam. Warm greetings from Perth in Western Australia. Welcome to my short talk on haplotype shuffling and SNP density crossover junctions within the MHC Class 1 and Class 2 genomic regions. My name is Jerzy Kulski, and I thank Dr. Paul Norman for his invitation to present this short video to your workshop. The 5 points that I would like to cover the most, but probably cannot get through all of them in this short time, are the following. 1. Homozygous cell lines generate MHC haplotypic phase genomic sequences. 2. MHC genomic sequences are highly polymorphic, but vary in SNP densities across the MHC. 3. The same haplotypes in individual homozygotes, or those inherited as identity by descent in populations, are highly conserved with relatively few or no SNP differences between them. 4. Comparisons between different haplotypes in heterozygotes have revealed distinct SNP profiles, haplotype shuffling, and regions of SNP crossover between SNP-rich and SNP-poor segments. 5. Repeat elements are an essential part of haplotype sequence analysis, especially in order to better understand the features and subtleties of genomic and transcriptomic regulation, as well as regions of ancestral recombination. These 5 points mostly stem from 3 publications, which I encourage you to access and read, because together they contain much more information that I can provide you with in this short talk. You can download the Kulski et al. Papers for free from the website of Frontiers in Genetics with the research topic Population Genomic Architecture. I would like to briefly acknowledge the initiatives and pioneering work and publications in regard to the MHC haplotypes by the following researchers and groups. These are Rugendo Cepellini, Chester Elper of the USA, Roger Dawkins et al. Of Perth in Australia, the MHC Sequencing Consortium, the MHC Haplotype Project of the Sanger Center, and the Norman et al. Haplotype Sequencing Initiative in the USA. In 1967, the Italian geneticist and HLA pioneer Rugendo Cepellini introduced the word haplotype. This was to describe alleles in the HLA system which segregated together in family studies. After 1967, the term haplotype has taken on some different meanings, but the MHC sequencing community generally knows how the term applies for the MHC genomic region. In 2017, Norman et al. Produced a landmark paper for 95 annotated homozygous MHC haplotype sequences that they generated from cell lines, including some of the 8 previously produced by the Sanger Center. What was interesting about this publication was, firstly, the new annotated information that the authors brought to the table about the MHC sequence variations. And secondly, they made the 95 MHC reference sequences available in the FASTA format for further analysis by other interested investigators. They genotyped the coding sequences at 163 loci, including the most polymorphic classical class 1 and class 2 genes, the non-classical HLA genes, and various other regulatory and structural genes. My aim in our 2 studies of 2021 was to use the 95 genomic reference sequences to identify and characterize particular indels of transposable elements to use as haplotypic lineage markers, and also to elucidate the existence of SNP crossover loci at haplotype junctions in the DNA sequence alignments of different haplotypes. We use two simple internet bioinformatic computing methods for the sequence analysis. One, repeat masker for the detection of repeat elements using DNA sequences in the FASTA format, and two, PipMaker provided the sequence alignments in a PDF format to detect and visualize the position of SNPs, indels, and the repeat elements that were within the input sequences. The multi-PipMaker produced nice clear-cut nucleotide alignments in a PDF format that allowed the SNP to be examined manually in fine detail. In the figure to the left, you can see the sequence alignments and a few SNP differences in the line 1 and ALU sequences. PipMaker also produced a percentage identity plot showing the location of the repeat elements for orientation of the nucleotide alignments, shown on the right. Unfortunately, the percentage identity plots have been covered over by my inserted texts and coloured blocks. This methodology, together with the information and annotations provided by the Norman publication, was all that were required for our analyses. Our two published studies showed that, firstly, SNP density crossovers are associated with putative ancestral recombination sites that are widely spread across the MHC class 1 and class 2 genomic regions. Secondly, the genomic sequences of MHC homozygous cell lines are useful for analysing haplotype blocks, ancestral haplotypic landscapes and markers, and SNP density crossover junctions. Thirdly, substantial haplotype shuffling exists between different polymorphic blocks and confirms the presence of numerous putative ancestral recombination sites across the MHC genomic regions. Now allow me to take you on a quick tour of MHC SNP profiles and SNP crossover regions between the SNP forests and deserts that demarcate MHC haplotype blocks. The first SNP profile plots that I saw between two different aligned MHC haplotype sequences were reported by Gordieri et al. In 1999 and 2000 using the Mann and Boleth homozygous cell lines. 22 years ago, they then looked quite astonishing. Reporting from the telomeric end on the right to the centromeric end on the left of the figure, it can be seen that most of the SNP density is around the HLA-A, B, and DQA1 genes in this very early published plot. Later on, Stewart et al. In 2004 and Traherne et al. In 2006 of the Sanger MHC haplotype project published more sophisticated SNP diversity plots using fully sequenced MHC haplotypes of the cell lines such as PGF, COX, and QBL. In the figure showing the SNP profiles, the MHC class 1 region is on the left and the class 2 region is on the right. Norman et al. Also published a SNP plot for their 95 reference haplotypes. The trend of the overall SNP profile remained the same as the Gordieri and Traherne publications, with major SNP peaks occurring within the alpha block around HLA-A, the beta block around HLA C and B, and the delta block around HLA DRB1, DQA1, and DQB1. A smaller significant peak is also seen around HLA DPB1 within the epsilon block. In 2006, Smith et al. Produced a variation on the theme that included a SNP diversity plot of 19 phased chromosomes with the same conserved Caucasian haplotype, known as 8.1 or the 8.1 B8DR3 haplotype. You can see the typical SNP profile in green between different haplotypes, and the flat red line between haplotypes identical in sequence from HLA-F to HLA-DQ. Thereafter, there is a SNP crossover junction from a SNP poor to a SNP-rich region within the loci of DP. It is evident from all of these plots from different research groups that the MHC SNP profile between different haplotypes is very distinctive and reproducible. But the trend of the overall SNP profiles remains the same as the Gordieri et al. Publications. Most of the SNP diversity occurs in the alpha, beta, and delta blocks. This fact was known for a long time, and that's why the Dawkins group named these HLA genomic regions polymorphic frozen blocks. The Traherne publication of 2006 shows a definite SNP recombination crossover point between COX and QBL haplotypes in the HLA-DR region at a location between the HLA-DRB3 and HLA-DRB2. That is, while the COX and QBL sequences are markedly different on the left-hand side of the figure, they are almost identical on the right-hand side of the figure, centromeric of the HLA-DRB3 gene and in the region labelled as the SNP desert. The Kulski et al. Studies of 2021 used the reference sequences of Norman et al. To examine crossovers in the MHC class 1 and class 2 regions. The following are a few examples of the comparative sequence analyses between the same or different HLA class 2 haplotype pairs. In example A, the SNP profile plots in red represent the peaks and troughs of SNP differences across the entire MHC class 2 region between two different haplotypes. In example B, at the bottom of the figure, the SNP profile between the same conserved sequences have few or no SNPs across the entire MHC class 2 region. The SNP desert is represented in the figure as a continuous thick yellow line. In the next example, in figures C and D, 3 different SNP density crossover regions were identified between the different haplotypes. In figure C, the alleles from DRB1 to MTCO3P1 were different at each locus between the two aligned sequences. However, a SNP desert was detected at the DQB3 to the DQB2 loci. And then SNPs again emerged thereafter until another SNP desert was detected in the DP gene region. In summary, we analysed the sequence alignment of 136 haplotype pairs and found a total of 171 SNP crossover junctions across the MHC class 2 genomic region. The number of single crossover junctions per haplotype pair was 81, and there were 90 haplotype pairs that had 2 or more crossover junctions across the MHC class II genomic region. These crossover regions are assumed to be ancestral recombination sites involved with haplotype shuffling. The main message of my 2 published papers in Frontiers in Genetics is that SNPs can be used as markers for elucidating putative recombination crossover regions by judicious comparisons between different MHC haplotypes. However, we still need many more MHC haplotypes elucidated from different ethnic populations. What we've learned from our studies and those of others is that haplotypes are genetically important and informative, but still highly understudied and possibly even undervalued. So finally, please enjoy your workshop and keep on haplotyping. Thank you and goodbye.